Thanks again, Kelly, and thanks again the committee members for allowing me to speak and share our research. For this talk, I will limit, I'll talk about mostly the Batten disease work that we have been doing and not talk about the Parkinson's research that we're doing. So once again, I'm Indranil Basak. I work with Associate Professor Stephanie Hughes. Our lab focuses on a childhood neurodegenerative disease called Batten disease. And I'll talk about that, like what research we do in the next few slides. But before that, I want to draw your attention towards a video, which is on the next slide.
We've got this little girl who was running around and shooting hoops. She loves still ball games, soccer, and things. So yeah, this little girl who's just losing all her abilities to do everything. It's heartbreaking. She started having seizures after about the fourth lot of medications. They said, well, maybe it's not epilepsy. We had another appointment at the hospital and we found out that she has late infantile Batten disease. She will lose most of her abilities. She'll probably be blind. She definitely won't be walking. She'll be peg fed. And if we get through the winters and pneumonia doesn't get her, she'll just be vegetative. There won't be a cure for Katie in her lifetime. It's fatal. So a lot of the children go 8 to 10 years, but it'd be really nice to think we're helping for the future and maybe helping some other children.
So that story is about the little girl called Katie Archer. She was diagnosed with one form of the Batten disease called CLN6 Batten disease. And unfortunately and sadly, she did not survive and she passed away at the age of 10. As you already heard, that there are no cures for any form of the disease. And that's what the motivation that brings us back to the lab and to get this thing up and running, get our research up and running and to find something that can provide better life for these kids and their families. So once again, a very brief overview of Batten disease. There are symptoms that include blindness, seizures, hallucination, motor problems, learning deficits, and inability to walk, talk, swallow, and feed. There are 13 different forms of this disease. Each is caused by mutations in 13 different genes. These are called CLN genes. For this talk, I will focus on CLN5 that we have been studying in this lab for several years. And if you ask us, like, how would you define like Batten disease briefly, we always say it's kind of like epilepsy, Alzheimer's disease, Parkinson's disease, all three put together in children. Just imagine how horrible that can be, right? Not just for the kids, but also for their parents and the families and the community. So as I mentioned, there are no cures for this any form of the disease. There is only one treatment for one form of the disease, which is an enzyme replacement therapy. However, there are some clinical trials going on. One of them is for a gene therapy. Recently, at the beginning of this year, it went into clinical trial. This is for CLN5, and our supervisor, Associate Professor Stephanie Hughes, she has contributed a lot towards the development of this gene therapy. When this gene therapy went into clinical trial, we did not know what the protein function is. Like, what does CLN5 do? So CLN5 gene, it encodes for a CLN5 protein, which is a lysosomal protein, but without any known function until this year. So we wanted to know more about what happens in the neurons, which are deficient of CLN5 protein. That's why we developed an iPSC, or induced pluripotent stem cell derived neuronal model to mimic Batten disease, or CLN5 Batten disease, so that we can get answers to what's going wrong, and we can contribute towards finding a better cure and finding ultimate care for the patients. So what we use is, as I mentioned, iPSCs, and our model iPSCs basically have a transcription factor called neurogenin 2. which is like expressed under a doxycycline promoter. So once you expose the iPSCs to doxycycline, the neurogenin 2 expresses, and it drives the differentiation of the iPSCs towards pure cortical-like glutamatergic neuron at the end of day 14 to between day 14 to day 21. That's how they look like at between day 14 and day 21. The positive bits of, or the advantage of this system is it's a rapid system. You can grow them really easily. It's not that hard to grow them. And this model can be used to mimic other diseases, brain diseases in a dish. You can look at single cells, you can look at bulk neurons, great for microscopy and bucket biochemistry, and also an ideal model for high throughput drug screening. So what we did was we used CRISPR-I to inhibit CLN5 in these neurons. So as you can see, we have used 3 different guide RNAs to knock down CLN5 from our neurons. So guide RNA number 2 and 3 didn't show any protein, whereas number 1 showed some, like a bit of the protein left over. So the knockdown was pretty good. So it was the time to characterize the phenotypes of resulting from these CLN5 deficient neurons. Now, from previous studies, not only from our lab, but also from previous other labs, we know that Batten disease is considered as, or is classified as a lysosomal storage disorder. So there are definite evidences that Batten disease, or especially CLN5 Batten disease, show lysosomal defects. Now, what are lysosomes? So lysosomes are basically janitors of the cells. So they keep the cells clean, they degrade the cellular waste, they recycle the cellular waste, and they keep the cells healthy and working. But if the lysosomes are unhappy, what we get is different types of diseases. And one of the classes of diseases is neurodegenerative diseases. You will probably see or have seen papers showing lysosomal dysfunction in more common disorders like Parkinson's and Alzheimer's disease. So in neurons, lysosomes are even more important because lysosomes significantly contribute towards maintaining synaptic activity and development, neuronal health, and lysosomes travel from one end of the neuron to another end. So a recent paper showed that RNA globules or molecules, they actually hitchhike on lysosomes to go from the cell body to axons or dendrites. That's why lysosomes need to move throughout the neuron, not just only to degrade cellular waste, but also transport cargo. And of course, lysosomes enable autophagy. So the first phenotype that we checked in these CLN5 deficient neurons that we generated using CRISPR-I model in the iPSC-derived neurons was to check how the lysosomes look like. So the first thing that we tested was lysosomal acidity or acidic organelles. Now, lysosomes are acidic organelles, and it's basically, lysosomes are basically enzyme-filled organelles where the enzymes can chop all the cellular waste. But if the acidity goes up, or if the lysosomes are not acidic, if the lysosomal environment is not acidic, the enzymes will stop working. So the first thing we tested was whether there are changes in lysosomal acidity or acidic organelles in CLN5 deficient neurons. And the green dots that you are seeing are nothing but acidic organelles measured by lysotracker. And in the CLN5 neurons, you can see like there are way less number of acidic organelles. The next bit we tested was to check like cathepsin B activity. This is an enzyme, a protease, which resides in the lysosomes. We noticed that with CLN5 knockdown, there is a decrease in cathepsin B activity. Now, remember, I mentioned that lysosomes need to move within the neurons. So the next phenotype that we noticed was movement problems in the CLN5 neuron. So the white dots that are on the left panel, these are nothing but lysosomes. So you will see there are some white dots moving around back and forth and up, right and left. And there are some lysosomes which are static. So that's normal. But in CLN5 deficient neurons, what happens is, if I may draw your attention towards these arrows, you will see these lysosomes, they kind of like start moving, but they just don't know where they're going, and they get back to where they start from. In a minute, here we go. So it goes front and then it goes back again. So we see these kind of defects in the CLN5 deficient neurons, which means that the lysosomes are unable to move. And that means like there will be more toxic materials accumulated in the system, in the neurons, which can lead to neurotoxicity. So when we quantified these movements with chymograph, what we found was these zigzag lines, which are basically the lysosomes going back and forth in the control neurons, whereas the straight lines are the lysosomes that can't move, they're static in the CLN5 deficient neurons. Apart from the lysosomal defects, we also noticed mitochondrial defects, which has been also shown in other cell types deficient of CLN5. And we also noted defects in autophagy, again, something that we expected, and some accumulation of key proteins involved in other diseases as well. So what's happening in the cells? That was the main question for us. So to get a better look into the global changes in the cells, we took a multi-omic approach. So we looked at changes in DNA methylation, transcriptomic changes, and proteomic changes. And I'm going to show you a few preliminary data that we have found that is very interesting when it comes to CLN5 deficient neurons. So from the methylation data, we found that there were like a set of genes which were hypomethylated. Now, in general terms, hypomethylation means decrease in methylation, which leads to increase in gene expression. Again, generally there are exceptions, but that's the general concept. So these genes that showed hypomethylation were actually involved in cell proliferation. That is, increase in gene expression of the genes of these genes involved in cell proliferation. And a similar effect we also found in the CLN5 knockdown neurons from our transcriptomic data. So we saw an increase in gene expressions in CLN5 knockdown neurons, which are involved in cell proliferation. So kind of our DNA methylation data complements our transcriptomic data, but we still don't know why there is an increase in cell proliferation when CLN5 is knocked down, especially when CLN5 being in the lysosomes. We still don't know the answer. However, we are not the only ones to see this. In 2017, there was a paper in mouse where CLN5 was knocked out, and they noticed that there was altered neurogenesis, in fact, increased neurogenesis. And there is another form of Batten disease, the CLN7 Batten disease, where in a social amoeba model, it has been shown that loss of this protein leads to increase in cell proliferation and neurogenesis. So initially we looked at the number of cells being how they are dividing at the iPSC stage, as well as at the induction and differentiation stage of the neurons. And we found that there was an increase in the number of cells that are going ahead, at the iPSC stage, as well as the induction phase in the CLN5 deficient neurons. We also have some preliminary data that confirms or validates our finding using Ki67 staining and an organoid model of CLN5. So that's something we are working on currently. We don't know again why this is happening, but we are trying to find what can be the reason behind it. So that's the upregulation. When we went back to the transcriptomic data, we found that there was a downregulation of ion transport as a whole process. So one of our students, Josh Clegg, he found that there are multiple potassium channels that are downregulated in CLN5 deficient neurons. There are other channels as well, like the zinc transporters and glutamate transporters, which I'm not going to talk about today. So what he did, what Josh did during his honors project this year, was he chose KCNH4 based on the literature, because this is a gene that is involved in epilepsy, and he knocked it down, and he found that there is an increase in cell death when you knock down or when you inhibit KCNH4 in the neurons. And he also did a lysotracker assay, again, looking at lysosomal acidic organelles. And what he found was there was a trend of decrease in lysotracker signal when the KCNH4 was inhibited. So that's all good. And we are also looking at other transporters, and we are collaborating with Professor Anthony White and trying to look at what other ion channel or what other ions are not normal in the CLN5 neurons. Another interesting fact from the transcriptomic data that came out was long non-coding RNAs. That's something that has never been shown in any form of Batten disease. So these two students, Alex, who is again another honor student, and Kirsten, who is a PhD student, so they started working on link RNAs or long non-coding RNAs, and we found that there are a few link RNAs that are upregulated and some are downregulated when CLN5 is missing. So Alex started working on Neuralink 2, and he found that Neuralink 2 is involved in several cancers. There are several papers. There are a few papers on Neuralink 2 and how deficiency of Neuralink 2 can lead to neurodevelopmental defects, especially in autism spectrum disorders. And when he started looking at these papers, they were not like they were just trying to either knock down or overexpress this Neuralink 2. However, what he found was there are 43 different transcript variants of this long non-coding RNA. And in our neurons, there are several of those variants that are expressed. So it was really difficult to pick primers, design primers, and design guide RNAs to specifically knock down some of the specific highly expressed transcript variants. He managed to do that by inhibiting 2 of the transcript variants, and what he found was there was an increase in lysotracker signal when he knocked down or inhibited these 2 isoforms, or not isoforms, variants of this NL2 or Neuralink 2, which kind of complements with our previous observation that CLN5 decrease leads to decrease in lysotracker signal and increase in Neuralink 2. But when you inhibit Neuralink 2, there's an increase in lysotracker signal. So moving ahead, this can be a really good target for therapeutic potential. So the next step will be to test the effect of the knockdown of this Neuralink in the CLN5 deficient neurons. Moving on, the next bit was to look at the proteomic data that we did. We detected more than 3,000 proteins, out of which there were like 160 proteins upregulated in the CLN5 deficient neurons versus the healthy neurons, and they were involved in cytosolic microtubule organization, neurodegeneration pathways, and ubiquitin proteasomal system. The UPS proteins, we expected that because the lysosomes are down, so cellular waste are being accumulated. So the UPS is trying to do its bit so that the stress on the cells are minimized. So kind of like a compensatory mechanism. And then we also looked at some of the downregulated proteins, and they were involved in 2 main clusters. I'm not trying to draw your attention to the details of the string diagram, but you can see like there are 2 specific clusters. One is involved in intracellular transport and the other one in translation. So when we started looking into these 2 pathways, and we started looking into what are the proteins, we found out what are these proteins involved in more specifically. And what we found was these proteins are the ones that are decreased in CLN5 inhibited cells are actually involved in ion transport, which is something which we have seen in from our transcriptomic data. So that complements our transcriptomic data. We also found proteins that are inhibited or that are downregulated in CLN5 deficient cells that are involved in translation, protein targeting to membrane and organelle, protein localization, and synaptic transmission. So that's pretty much about the new results that we have seen so far. So to summarize, in normal or healthy neurons, we have CLN5, great, the cells are happy, but when you knock it down, you see an increase in cell proliferation and UPS system. And then you see also deficiency in or decrease in acidic organelles, problems with lysosomal movement, which is key to maintain neuronal health, decrease in translation, problems with protein trafficking to different organelles and the membrane, and ion transport. Now, when we were doing this multi-omic study, a paper came out from Robert Steinfeld's lab showing that CLN5 is actually an S depamethylases. So that's great. If CLN5 is inhibited or blocked, what happens is there will be hyperpamethylation. Now, let me remind you, pamethylation is a very important post-translational modification that regulate trafficking of proteins, neural synaptic transmission. That means when CLN5 is blocked, there is hyperpamethylation, which could lead to defective protein trafficking. That's something we have seen from our proteomic data, and also excitotoxicity via depletion of potassium channels. Again, something that we have seen in our cells. So that's where we are standing right now. We know that CLN5 Batten disease, or any of the Batten disease, it's not just limited to neurons, and there are other cells in the brain, like non-neuronal cells, that are affected in Batten disease. And one of the cell types that we are studying currently is astrocytes, so one of the glial cells. So what we did was we developed these human astrocyte model from iPSCs, again, following a recent paper. And we used CRISPR-I and CRISPR-A to inhibit and activate CLN5 in these cells. We haven't characterized these cells yet, but that's something we're going to do in the next few months. We also, as I mentioned briefly before, that we also have an organoid model, which is deficient of CLN5. And what we are doing at this point is characterizing, or we will be characterizing, this brain organoid model. So that's where we are, and that's what we have been doing with all these different models. We hope that all these models and all the multi-omics analysis, these information give rise to valuable finding what happens in the cells when we don't have CLN5, and that could actually help in the future to make better judgment for developing treatment strategies for CLN5 Batten, is to help kids like Katie with specifically CLN5 mutations. So that's pretty much from me. And finally, the most important slide, thanks to our group leader, Associate Professor Stephanie Hughes, for her immense support and all the experience bringing with her and sharing with us. To all of our lab members who have been helping throughout the whole project, to all the families of the patients who have supported us, to our collaborators. Dr. Michael Ward he developed the Neurogen iPSC system, and to all of the funding agencies without which this work wouldn't have been possible. So that's pretty much from me. Thank you very much.